I try to make it an interactive presentation, so you're gonna need your cell phone with you to see like if we can just stay up as I talk through. It should take about 30 minutes for the presentation, and then at the end of the presentation, we're gonna talk in group. Okay, so I am a postdoctoral fellow in Professor Itai Budin lab at the University of California in San Diego in the chemistry and biochemistry department. And today I'm going to tell you about the role of lipids in evolutionary adaptation. So here's a little picture of how we draw lipids chemically in the lab. Okay, so before I jump into the sciences, I want to tell you a little bit about my journey into becoming a scientist, which is pretty unusual for people. I was born and raised in Ecuador, in Quito, but I fell in love with sciences going with my mother to the Amazon rainforest. She's a doctor, she's an MD, and she was working for an American organization that was sending medicine and medical attention to remote tribes of the Amazon when I was little. And she got to take me and my three other sisters with her to help her with things. The amazing thing is that I was able to be in the room with her when she was taking care of patients. So I was exposed to like different infections and different crazy types of diseases when I was very little. And I just became so fascinated about the human body. So we relocated with my family to New Mexico in 2008, and I finished my last year of high school in a little school called Española Valley High School at Northern New Mexico And then I moved to a bigger city called Albuquerque, New Mexico, where I finished my bachelor's degree in biology. And when I was there, I had the opportunity to do an internship in Seattle at the University of Washington in the Genome Sciences Department, where I was able to co-author a publication with a professor there. So I fell in love with genome sciences, and I moved there to continue with my PhD. I don't know. Okay, and and here's a picture of me through my PhD. I work with yeast. This is a picture of yeast. And a little bit later, I work with the fruit fly or Drosophila. And for both of them, I work with genetics and genomics. So yeah, after I finished my PhD, I moved to Canada. I accepted a position in a company that is making meat in the lab. And they're using the fruit fly in order to make transgenic proteins. So we end up making food that is completely without the use of other animals. So working in that company, I was the senior research scientist there. I was in charge of setting up the use of Drosophila or the fruit fly in making these. I realized that our knowledge in terms of lipids was lacking, and we are already moving really fast in this field of making meat in the lab. So I decided to go back to academics and actually start studying lipids, fats. And that's what it brought me here to San Diego to work with Professor Itai Budin, who works in lipids. Okay, and that is what I'm gonna be telling you about today in this presentation. I'm gonna be telling you about what lipids are and how they're linked to evolutionary adaptation. That's gonna be the first part of the workshop. And then for the second part of the workshop, we're gonna be talking about how to bring this knowledge into the classrooms. So at the end, there are going to be these 4 things that we're going to be working on. So I just want to ask you to keep in mind these things as I go through the presentation, especially trying to catch one major main concept that you would like to bring into the classroom. So like at the end, we can talk about it. Okay? Okay, so I'm going to start now with the sciences part. If you live in the city, it's really hard to come across the amazing biological diversity that our planet has. These pictures here were taken actually in San Diego during one of the tide pools that they form here. And you can see these amazing animals that we don't really come across very often. And they're very different from other types of animals found in other places, such as these birds or frogs or this lion. And what is amazing about these is that even though they're so diverse and so different in terms of sizes, in terms of where they live, the temperatures that they survive at, there are 3 major building blocks that made them who they are, which are the DNA, proteins, and lipids. And at that level, those building blocks are very similar to each other. So they're very limited. So for me, it's just amazing to think about how such a limited amount of of components can make such different diversity in our planet. So today I would love to talk to you about DNA, but we're not going to talk about that. We're going to be talking about lipids, which is what I'm studying right now. So to start, I want to invite you to go into this website. We're going to make it a little bit interactive here. Can you see my other screen? So we're going to be going into this website called menti.com, and you can use the code 81996877, and it should pop out this question here. What comes to mind when you hear the word lipid? And you can share your responses, and they should start popping out here. So if you don't know, if nothing comes up when you think about lipid, that's completely fine. You can also say, I don't know, or nothing. Okay, awesome. So membranes, hydrocarbon, fatty acids, great. Fats. Awesome, yes. So if you didn't have a chance to enter an answer, it's okay. There's gonna be more activities like this that you can enter. So you can actually keep this website open in your phone if you like to. Phospholipid bolsters, wow, awesome. Yeah, so one of the things that lipids are known for is to be components, major components of the cellular membrane. So usually in biology textbooks, you see pictures like these in which lipids are drawn like these little bubbles with two little tails here. And they're usually surrounding components inside of the cell. So lipids are very important to separate different environments. They separate an outside from an inside, which is important to keep homeostasis inside the cell. They're very important for selection and filtering of molecules. Lipids usually allow water molecules or CO2 to go through the membrane, but they don't allow big molecules to pass through. They're also important for signaling, which is something people don't think about a lot, and mainly because they embed proteins and receptors within the membrane. So depending on how compact those lipids are bounded to each other, they might allow for flexibility for proteins and ion channels to send out the signal down to the cell. And lipids, finally, are very important for adaptation, which is what I'm going to be talking about today. So even though lipids in biology textbooks are drawn in this way, they're very homogeneous, very similar to each other, that's actually pretty far from the truth. Lipids are actually, the cellular membrane is actually made of a huge number of different lipids, and they're very mobile. So if you are to tag one lipid here, like this one that I'm showing in this little cartoon, that lipid would be moving around the cellular membrane, and it would also be very different from other lipids that make the membrane. The diversity of lipids come from the chemical structure of the lipid. So if we look at the actual molecules that make the lipids, lipids are usually made of a head group, made of a phosphate group, and a glycerol backbone, which is the little circle that people draw in textbooks. And then they're made of a fatty acid tails, which is the little tails that they draw in textbooks. The diversity of lipids based on the head groups can change the curvature of the membrane. So there are some lipids that have head groups that are the same size as the chains that they have, making a little square in the membrane. And those lipids tend to keep a membrane pretty straight. However, there are other lipids that have bigger head groups, such as these, if they have a serine in here bound to the phosphate group. And what that does is it bends the membrane inward because the heads are bigger than the tails. There are other types of lipids in which the head is pretty small, such as this ethanolamine. And in this case, if membranes are composed of that type of lipid, the membrane curvature is going to go to the other side. Another way of increasing the diversity of lipids is by increasing the amount of unsaturation of the tail. So now we're going to be looking at this part here. The tail is made of a carbon chain, like somebody answered in the poll before. And those carbons are usually bound to the max number of hydrogens that they can. And when that happens, that chain is considered to be saturated. However, sometimes those carbons are bound by two double bonds, losing one hydrogen each one. And in that case, that chain is considered to be monounsaturated. And it can happen that some of the chains can have multiple double bonds, in which case we call that to be polyunsaturated chain. And thinking of lipids in this way is important when we think about cold adaptation, because that is what it allows the lipids to keep a specific type of phase at a specific temperature. So this is easy to visualize when we think about butter. Butter is made of lipids. When you warm up butter, when you increase the temperature, butter becomes liquid. And that is because the lipids start losing contraction with each other and they become more fluid. As temperature goes down, those lipids, because of thermodynamics, the lipids tend to compact with each other, making butter a little bit, making butter more solid, solidifying butter. Now, if you introduce one double bond into the chain of those lipids, that is going to bend the chain in a specific way, making it harder for those tails to just compact with each other, and giving that lipid a specific melting temperature or phase transition at which they become solid. Now, if you introduce two double bonds, that's going to introduce even a stronger bending into the lipid, now decreasing the melting temperature or phase transition to -5 degrees. And now if you introduce an extra double bond here, you can see that the melting temperature goes even lower to -11 degrees because the chain bends even more. Now,
Claudia,
yes.
Sorry, just quick question since you invited us to ask questions. So you have that, the melting temp is the TM, right? So you can think of it as that. So basically, when you get to that number, that's when it becomes a solid. If you're lowering and lowering and lowering and lowering the temperature, that's when it becomes solid.
Correct. Yes, correct. Yes, yes,
right.
Correct. And it's kind of tricky to think about because it's melting temperature, but it's when it transitions to the next phase, which is solid. Yeah.
Cool.
Yeah, correct. Thanks, Alec. Okay, so I want to introduce here a few terms that I'm going to use for later on in the presentation. So for one double bond, I'm going to use the term MUFAS, which stands for monounsaturated fatty acids. For two double bonds, I'm going to use the word DUFAS for diunsaturated fatty acids. And for three or more double bonds, I'm going to use the word PUFAS for polyunsaturated fatty acids. And again, feel free to make me remind you of these terms later on if we come across this. Okay, so the diversity of lipids is really important to think about in terms of what makes an organism, because if you imagine if an organism is made only of one type of lipid, let's say in this example of the lipid PE18,3, which has a melting temperature of -30 degrees, that organism in our world would be pretty much melted. On the other hand, if you have an organism that is made of another type of lipid, P18,1, which has a melting temperature of 71 degrees, that organism wouldn't have any type of mobility. It would be very, very solid. And in the lab, we have actually found examples like this. These are photos from a paper that is under publication. It's actually Itai's work with another person from the lab. And here I'm showing you a type of organism that lives very deep in the ocean. The name of that organism is called a ctenophore or comb jelly. And this organism lives at very high pressure and cold temperature. And the lipids of these organisms are very different from the lipids of organisms that live up here in the surface. And actually, when you bring this organism up to the surface, like here in the picture, you can see that that organism starts breaking apart completely because the lipids are losing that compaction. So it's really cool. Now, if you think about the human body in terms of diversity, you can start appreciating the types of lipids that our body needs to have access to, because different organs live in microenvironments in our body. So the stomach has a very high pH, or the tendons or the junctions in our bones are under different type of tension. So those organs are going to require different type of lipids as organs, let's say in the heart or lungs. And we have a little bit more of information in terms of biology on how lipids are distributed in organisms along the body, based on information that we have from organisms in the wild and how they have adapted to cold temperatures. So deer, for example, deer is made of different types of lipids. The tip of the legs that are in touch with the cold are usually made mostly of polyunsaturated fatty acids, so the ones that have a lot of unsaturation in their tails. While in the part of the legs, the majority of lipids that make up this upper part of the leg that is not in touch with a cold temperature, those are monounsaturated fatty acids. So we already have an understanding of the link between these unsaturations and cold adaptation. But in terms of the mechanisms, we don't really understand how that happens. How exactly is it that cells know what type of lipid they need under what type of environment? So to answer this question mechanistically on how, I am turning my attention to the little fruit fly, Drosophila melanogaster, which is what I train on during my PhD. So Drosophila melanogaster is the little fruit fly that you guys find in the kitchen. And Drosophila is able to outsurvive in places where the winters get pretty cold. So here I'm showing you a map of where this fly is found, which includes places such as Canada and Russia, which experience very cold winters. And Drosophila is able to survive through the winter. Now it's interesting in terms of Drosophila because Drosophila in itself doesn't have the ability of make these polyunsaturated chains. It only has one enzyme that introduces one double bond, but it doesn't have the other enzymes that introduce the two double bonds or three double bonds. So we have a hypothesis that Drosophila actually uses diet. The main diet of Drosophila is yeast to obtain those polyunsaturated or diunsaturated fatty acids. Some yeast organisms can make those. There are certain yeast organisms that cannot make, similar to Drosophila. So in order to test our hypothesis, with a collaboration with Emily Berman at the Genelia Institute, we are working on this project. Emily went out to the field and she by hand collected flies in this vineyard during different months of the year, where the temperature varied between 30 degrees and 0 degrees. So she went out in June, August, September, October, November, December, and she collected flies that were on those places. Then she took those flies into the lab, and then she washed those flies and collected the yeast that were associated with those flies. And we saw that flies were associated with yeast that didn't produce any PUFA. So these are the pink colors that I'm showing you here. The majority of the yeast that were associated with flies didn't produce any PUFAs in the month of July when it was warm. But as the temperature got colder, such as in November, the amount of yeast that produce PUFA increased in those flies, giving us an idea that there is a connection between PUFA producing yeast and temperature. So I took this into the lab a little bit further, and I fed flies with two different types of diets. So these are the same flies in both tubes. The only difference between the flies is that they were fed on different foods. So here on the left, these flies were fed with the PUFA diet, which has the 3 unsaturations here. And the flies on the right were fed with a MUFA diet, which only has 1 unsaturation. These flies were put at 10 degrees, which is pretty cold for flies, for 24 hours. And you can see that the movement of these flies is affected. The flies fed with PUFA diet are able to move faster after I tap them down. It's very obvious. Okay, so I'm gonna talk a little bit about how we go from there into understanding how this happened. So for this, I broke out the talk into 4 different topics. I'm gonna start talking about the behavioral assays that we use. So in order to quantify the activity of flies in the cold based on the food that they are fed, I use this activity assay called Drosophila Activity Monitor, which allows me to use flies that were fed on different diets, so PUFA, DUFAs, or MUFAs, and load them into like little tubes. And then there is a beam that crosses through those tubes. And every time that the fly crosses that beam, that sends a signal into the machine and counts as a 1 count of activity of that fly. So if the fly is crossing the beam multiple times, then it's counting as more activity or not. Now, there is another type of behavioral assay that I use as well for food selection, but I'm gonna touch on that a little bit later. I just wanted to make the link here. So in terms of this Drosophila activity monitor that allows me to quantify the amount of times that the fly crosses the beam, I'm gonna show you data here, actual data. At 25 degrees, the flies fed on PUFAs had this amount, about like 500 counts, and the flies fed on MUFAs had a little bit lower, less than that, but it's not a lot less. When I put the flies at 8 degrees, however, which is pretty cold for the flies fed on MUFAs lose activity completely. The flies fed on DUFAs have a little bit higher of activity, but it's not a lot. And flies fed on PUFAs have actually a lot more activity than the other ones. This drastic difference is even easier to see when we normalize the data to PUFA. So let's say that PUFA activity is 100%. You can see that if we keep that at 100%, we see the difference of DUFAs and MUFAs, in which those actually decrease tremendously when we decrease the temperature from 20 to 8 degrees. Okay? And again, just a reminder here, the blue one is connected to this PUFA diet, which has the 3 double bonds. The green one is the DUFA diet, which has 2 double bonds, making it very obvious that it's something about the PUFA diet that is making the flies move in colder temperatures. Okay, so with that, let's go back to our activity room. Let's see if I can.
And can you share there's the code. It's up at the top, right?
Yes, it's at the top. So the next question is going to be, what type of diet allows flies to move in the cold? Highest activity as 1 and lowest activity as 3. And I think there is one person who wants to join, Alec.
Oh, Julian, yeah.
Okay, great. So we are having some answers here. And I don't know if everyone answered, but this is correct. So it's the PUFA diet that is allowing flies to move the most in the cold, and then the DUFAs follow, and the MUFAs give them very little activity. That is correct. Thanks for paying attention, you guys. Okay, so next I'm going to talk about the second assay that we use in order to understand the picture even more, and that is by using lipidomics data. So with lipidomics data, we take the same flies fed on the different diets. We smash the flies in a little tube, and then we just collect the lipids of those flies, and we send those out for analysis. They usually use machines that look like this for in the outside, but then in the inside, they have such a complex system, which I'm not going to talk in detail here. But pretty much, very briefly, this system allows you to take lipids to shoot it through here, and it's going to separate those lipids based on the mass that those lipids have. And the end result is a readout of different types of lipids, in which I'm able to see different head groups that we talked about before, or different amounts of unsaturation. So I'm going to show you here real data that it's something that we're working on, and we're going to send out this for publication. So I fed these flies on a PUFA diet, which again is many double bonds, or a DUFA diet, which is 2 double bonds. And we're going to be analyzing here only the head groups, so only the blue things that we usually see in lipids. And what I want you guys to pay attention here or to focus on is in the difference between the two colors, more than comparing them between PC and PE, because that is a different type of analysis. So between the two colors, you see that there is no difference. The flies fed on PUFA diet or DUFA diet have the same amount of the different head groups here. So that is not a big major difference that we're seeing. Now, if we focus only on the flies that were fed PUFA diets, and we take only the data for these three different head groups, we're going to be now looking at the number of double bonds in those three head groups. And what we see is that while PC and PE don't have a large number of polyunsaturated fatty acids, this lipid class called PEO has a very large number of polyunsaturated fatty acid. So that is one finding that we did in the lab. So what is this PEO? PEO is also called plasmalogen and is a very peculiar type of lipid because in the tail, instead of having an ester bond that looks like this in one of the chains, it has what is known as a vinyl ether bond. And chemically, this vinyl ether bond allows this lipid to have antioxidant properties. So this lipid has been linked to Alzheimer's disease. This type of lipid has been linked to other neurodegenerative diseases. So it's a very interesting type of lipid. Another interesting thing is, like, if you remember back at the beginning of the presentation, I talked about these ctenophores that live at very cold environments. These ctenophores, the majority of the body is made of this type of lipid, which is very different from other types of organisms in our environment. So this was a really cool finding that we had in the lab. It looks like flies are using this type of plasmalogen in order to adapt to cold temperatures. Okay, so how do we go from there into keep drawing even a deeper picture on like how flies are adapting to cold temperatures? For that, we also turn our attention to genetics. And the way we do it is by looking at where the plasmalogen is synthesized in the cell. So the plasmalogen is made in this little organelle that usually in textbooks is drawn as like a little bubble here. It's called the peroxisome. And in the peroxisome, there is a lot of processes that happen, and I don't expect you guys to remember this at all. The major parts of this whole process is that usually fats that come from diet are transported into the peroxisome, and then there is different metabolic processes that happen here that make it so at the end product of this beta oxidation pathway is the plasmalogen lipid that we have here. So how this helps me understanding how it specifically flies adapt to the cold. It's by taking mutant flies that have mutations of different enzymes, let's say this ABCD transporter here, and seeing if I interrupt this enzyme here and interrupt the plasmalogen biosynthesis in certain ways, am I also affecting how flies are able or not able to move in the cold? So in that way, I'm able to understand the mechanistic processes that helps for cold adaptation. Okay, one more question, and this one is the last one. Let's see, this is the last question. Okay, so the question says, where is the plasmalogen synthesized in the cell? So we have the mitochondria, the peroxisome, and the endoplasmic reticulum. 3, 4, I don't know. I think that's pretty much who we have here. And that is correct. It's mainly synthesized in the peroxisome. Perfect. Thanks, guys, for paying attention. Okay, and finally, to understand a bigger picture of what's happening, what we also do in the lab is do a volatile analysis. So volatiles, and this is what I said before I was going to come back to, volatiles allow Drosophila or the fruit fly to identify what type of yeast is making either PUFAs or DUFAs. Volatiles are actually lipid-derived molecules, and those are the signals that the yeast are sending out to the fly and that the fly picks on in order to select what food they're gonna eat. So we use this machine called gas chromatography mass spectrometry, and I added this video here that I took this morning in the lab that allows me to have a sample in the tube that is being loaded here. And then there are some volatiles that come out from the sample and bind to this fiber. This fiber then moves and injects the volatiles into this column, and the volatiles are separated here, not based on on their mass, but based on the temperature that they're able to bind into this fiber. And in that way, I'm able to identify which volatiles are the yeast sending out to flies. And that is something that I'm currently working on. Okay, so that is pretty much what what the project is that I'm doing is. The big picture of this project, or where it can be taken even farther, is helping us understand how organisms will adapt to climate change that is happening in the world. So remember that this adaptation that Drosophila has with yeast has taken a lot of years to happen or to accommodate in such ways. But with climate change, maybe those organisms will have to start acquiring different types of adaptation in order to adapt to the cold.